Okay, we're going to be doing a couple of, one other thing before we get to the lecture today. You've been in this hall now for a few months, and I hope by now you've come to appreciate the aesthetics of it. But one thing that we haven't done is talked about the technology and the science behind it. And there's no better person to talk about this than Kamen Nikolov here. You may have seen him, and if you're ever at concerts here, he's just always got that thing in his ear like a Secret Service agent, and he's always fixing things, changing things, making sure that the hall is perfect for whatever performance is going on that night. I also want to mention that Kamen was my student, a graduate student in the master's program here at Sonoma State, and his focus was on educational technology. And he's obviously taken his expertise in technology and used it very well. So Kamen is going to give you all the ins and outs of Weil Hall, or mostly the ins of Weil Hall. So let me introduce Kamen Nikolov.
Thank you very much. Thank you very much. Good morning. Good morning. Can everybody hear me? Good. I want to welcome you to Weil Concert Hall. My name is Kamen Nikolov. I run the technical operations for the hall. But it takes a village to really make this hall sound good, and when you come to a concert, for you to be able to see everything and to hear everything. So I want you to turn around and say thank you to the two gentlemen in the back. That would be Dave Montijo and Jerry Ulig. Without those two guys right there, they're very shy. Nothing really sounds good. So welcome again to Weil Hall. I want to give you a brief history of why we have this hall here. This hall is modeled after Ozawa Hall in Tanglewood, and I know most of you have not been there. Ozawa Hall in Tanglewood is really almost an identical looking hall to what we have here, which, as you can see, there is a lot of wood. And most of you probably wonder, why do we have so much wood? Well, wood is still the best material for sound. For example, those of you who play guitar, your guitar is made out of wood. It's not steel, it's not glass, it's wood. Same with violins, same with pianos. So we have various types of woods, and I also want to mention that everything in this hall has a purpose. For example, if you look up, you will see the rails, but you probably will never notice that the distance between the rails, I have a little green laser pointer here, the distance between the rails is actually different. Now, when would you think we will put different distances between the rails? Well, there is an acoustic purpose and goal for it, and that is different frequencies will be able to pass through those rails, and some frequencies will bounce back. And the reason those spaces are different is to allow a variety of frequencies to mix in the hall so that you can hear me. Now, speaking of being able to hear me, I want you to look above me and notice those giant speakers we have. They were not here yesterday or the day before. They really got installed yesterday for a concert that we have tomorrow. So tomorrow we've got Lila Downs. I hope some of you come because we have really inexpensive tickets for you guys, or I think it might be even free, I'm not sure. But with that kind of a rig, you should be able to hear really, really well. And we're really happy. We're trying to get something like this on a permanent basis, although for now we're just getting them as needed. Speaking of that, how is the sound with the sound people there? Mr. Montijo, everybody, you can hear me good? Oh yeah, they're so happy. Good. Okay, a little bit about the seats that you are seated on. So there are slats in the back. Again, this is something that the acoustician felt was important. When sound travels through the seat, you can see a lot of the seats are not occupied. Well, when the seat is not occupied, sound will flow through the back of the seat and reach the next person, and the next person, and the next person. So in a way, we're allowing the sound to mix in the hall and be really acoustically superb. If you look up, or anywhere around actually, you will see that the windows have shades, and there are 55 of them. At the moment, all of the shades are down, which is a setting we use when we do amplified sound. So when you see somebody with a microphone, or if you see a speaker anywhere on stage, that means that we're amplifying sound. And when we amplify sound, that gets tricky. The sound can get muddy, it can get boomy. So to avoid that, we need to absorb some of the sound. Now, keep in mind that most halls around the world cannot change the acoustics, where this hall, we can change the acoustics. So earlier, what you heard was an acoustic performance, and usually for an acoustic performance, we would not amplify anything. It would be just the acoustic instrument. But for amplified performances, we keep the variable acoustics down. You can see we have seating all around. There are around 760 some seats where you guys are. And there are another 730 or so spread around. And behind me, that is a choir loft. So many pieces feature large choirs, like big symphonies. Pieces have a large choir, but most of them do not. So on many nights, those are just fantastic audience seats. Like when you sit there, you have a completely different perspective on the performance, because you see the performers, you see the conductor, and you're in the kitchen, sort of like. You see things that when you're seated where you are now, you don't see. Like little, behind the scenes things. Another thing I would like to point out, which is not apparent at the moment, you can see the stage is at its flat setting. Everything is really flat, but I don't know, those of you who are a little higher up might be able to see that there are these green lines that outline the 2 sections of risers that can come up, and 5 more risers come down. So we have a 5-step riser system. So when we have a symphony, everybody is kind of like in an amphitheater style seating. So it's just the typical orchestra configuration. So I'm sure a lot of this stuff is maybe not very important or relevant to you, but one thing that will be important and relevant to you is that we will be building an outdoor pavilion, the MasterCard Pavilion, where we're hoping to get the types of artists that you guys like. For example, I know you guys like stand-up comedy. So how many of you were here for Aziz Ansari? How many of you? Oh, so probably about 20%. Okay. So when you guys go to concerts, first of all, how many of you have bought tickets to go to like Oakland Arena or anywhere to see a show? Like recently, in the last 3 months? Yeah, okay. How about in the last 1 year? Last year, okay. Okay, so let me get some names of the artists you guys saw. So what did you guys see? Some names. Did you guys see Beyonce? Who did you guys see?
Wiz Khalifa.
Okay. Chili Peppers, okay. Childish Gambino, okay. Any other names? All right. So keep in mind, the people that you paid money to go see over there, in about maybe 3 years, will come here. They'll come to you. So just I want you to kind of wait in excitement of what we'll bring to you, because we will be putting a lot of popular shows that you guys will like. In fact, most of you are on the student government when you guys choose which kinds of acts you guys want to bring on the on-campus presents. I don't know how many of you are in student government, but those of you who are definitely have a say on which artists to come. Okay, I want to point out a few other interesting facts about the hall. So this hall features a rather unique setup because, I don't know if you guys have noticed, but behind the hall there is a pretty big lawn. And in that lawn we can accommodate about 4,500 people maybe. And what is quite unique is that the big wall behind you, that wall opens. So this summer will be our first season where we're going to have the concerts be inside and outside, like on an ongoing basis. We opened the doors for our grand opening with Alison Krauss, and that went really well. But basically we're going to do it this summer. Every concert is going to have those back wall, the doors, the big wall open. And that gets really nice because you have everybody inside watches the show, and we're going to put big screens on the building, and everybody on the lawn can bring a picnic basket and a blanket and enjoy the show. And usually the seats on the lawn are pretty inexpensive. In fact, sometimes they're free. So, okay, so I told you about the lawn. I told you about the upcoming MasterCard Pavilion. This is also the home of several presenting organizations. So we have, you heard the music department earlier, the various programs of the music department. We have a lot of concerts with the music department. We have the MasterCard series, so you've seen some of the names. A lot of classical performances, some jazz performances. Wynton Marsalis. How many of you like jazz? Okay, about 30%, okay. So Wynton Marsalis was here, I would say, what, 2 weeks ago, 3 weeks ago, with the Lincoln Center Jazz Orchestra. That was fantastic. They're so good. So then we have the Santa Rosa Symphony. So you see a lot of symphonic performances. We also have the San Francisco Symphony. They come and they perform. We've had several comedians. I don't recall the names, but probably you guys remember. And we also have the On-Campus Presents, which brings the comedians and other performers. Okay, let me see. I want to make sure I mention everything. Oh, one last thing, and I will return the podium to the instructor. But I want to mention that if you guys look down, there's one more element that is kind of important. If you look down, you're going to see a lot of black circles. And I know you're hoping that those are speakers, but they're not. So that's part of the air conditioning system. And usually, air conditioning system can be really loud and obnoxious, like in your houses, in your apartments. It's really loud. But here, the air conditioning is really quiet. And every one of you gets perfect temperature because there are hundreds and hundreds and hundreds of black air conditioning vents. So that's really important to keep sound in the hall clean and quiet and so that you can hear well. So that was a really important factor to the acousticians to make this hall a world-class hall. Another fact that you probably are not aware of is because we are now part of the MasterCard marketing program, people around the world are hearing about this hall. Like, for example, people in Japan and in Germany are seeing this hall in Sonoma County at Sonoma State University as a destination for a lot of concerts that are coming up. So this place is becoming well known around the world as we speak. And I think that's pretty much all I have to add about the hall.
I have one more question though.
Sure.
Isn't there something about the general shape of this hall? Isn't it called a shoebox? Yeah. Which is a pretty unique shape. Yeah, so I missed to mention the shape of the hall. So when you go to a theater, theaters can be any size, any shape, seashell type or round. Well, this is rectangular because the acousticians who have studied performance halls for classical music have determined that over the centuries, there is a shape of a hall that tends to work best for that kind of sound, and this is this rectangular, long shoebox design, which is modeled after really, there is a hall in Vienna, it's called Musikverein. So we are modeled after Musikverein in Vienna. So we're hoping to be among the top 5 in the world. Thanks, Carmen.
Yeah, you're welcome. Thank you.
Since this course is about change too, it makes me think of when I was growing up in the 50s and 60s and even as late as the 70s, when they built a concert hall, they never knew what the quality of the acoustics would be. It was total luck. So for instance, Lincoln Center, which is a fabulous set of concert halls in New York City, Alice Tully Hall, the main hall there, it was terrible. The acoustics were awful. And they didn't know until they actually opened it and started having concerts. So then over the next decade, they spent time and incredible amounts of money to put like baffles and different things up to make it work. On the other hand, when I was growing up in Los Angeles, Dorothy Chandler Pavilion, how many of you are from LA? So you know the Music Center in downtown LA, Dorothy Chandler Pavilion. That one happened to be really outstanding. That one has fantastic acoustics, and nobody really knew why. Although the Amundsen Theater, which is another one of the complex in the LA Music Center, that one was no good. So, but in the last 30 years or so, the science has really come a long way. So now they knew ahead of time that this would be really good. They just know if they put in the money and if you put in the wood and do all those things that it's actually going to work. So anyway, thank you, Common, for keeping it working, and everybody back there as well. Okay, so now we're moving into a new part of the course. We're going to be looking at ethics of science in particular. And I think it's a good segue from the lecture that you had on Monday. After the lecture, I was speaking to him, and we were talking about the whole issue of genetic testing. He said sometimes people will, if their baby has a runny nose and seems to have some problems, they want to get tested for I can't remember the name of the genetic problem that he was describing. And he said, it's not necessarily a good idea. He tries to tell them that unless they have a family history and a better likelihood of having this, it may not be a good idea to actually do the testing, because when you get tested, you might learn some stuff that could really cause tremendous amounts of worry. And also the issue of genetic testing think about this. What if genetic testing became something that anybody could require of you, like when you're going to get a job, right? So that they might look for a gene that would indicate a possibility of alcoholism, for example. I mean, think about the information that is contained in your genetic code, or just deciding whether you want to find out if you have something that a parent has. I have a niece whose father died of a genetic disease, a very rare genetic disease. And she's 28, and she has at this point decided not to find out if she has it, because sometimes that knowledge can be extremely damaging to you, but also damaging because then it's out there. So there are that's just around genetics. We're going to move into something a little different, but the segue from science and health and nutrition to ethics is, I think, a really smooth one. So I'm excited about introducing our provost, who's going to talk about this. So let me first just explain to you a little bit of how the structure is on the academic side of Sonoma State. There are 7 deans, and you've met all of them at this point, each of whom is in charge of an academic unit, like arts and humanities, or the library, or science and technology, and so on. 7 of them. So they're each in charge of an academic unit. Then there are 3 associate vice presidents who are in charge of some things across the university. I'm undergraduate studies, there's somebody else who's faculty, and somebody else who is academic programs. And we sort of comprise the leadership on the academic side of the university. And all of us report to one boss, and that is the provost of Sonoma State, provost and vice president for academic affairs, and that is Dr. Andrew Rogerson, who I'm going to introduce in a minute. So you all have to be on your absolutely best behavior, because keep in mind, he is my boss. Okay. There you go. He's actually really a great boss. So let me tell you a little bit about Provost Rogerson. He came to Sonoma State University after serving as Dean of Science and Mathematics at Fresno State. He was the Dean of Science at Marshall University and Associate Dean at the Oceanographic Center of Nova Southeastern University. He's also been faculty at the University of Toronto, the University of London, and the South Dakota School of Mines and Technology. His research interests are varied, and they include a lot of really little stuff, right? Is that right? The microbial ecology of protists, the biomineralization of silicon in diatoms, the taxonomy of naked amoeba. I thought amoeba never wear clothes, right? Naked amoeba, fecal contamination of beaches, the efficacy of ballast water treatment systems, and microbial pathogenicity. His research has been funded by federal or government agencies, including the National Oceanic and Atmospheric Administration, the U.S. Environmental Protection Agency, the National Science Foundation, and the National Environmental Research Council, that's in the United Kingdom. Provost Rogerson received his PhD in microbial ecology from the University of Stirling, Scotland in 1979. I also might add that he is the coolest provost ever to be at Sonoma State. He drives a motorcycle to and from campus every day, rain or shine, summer or winter. So when you see this guy leaving in his leather jacket and his big old helmet, you'll know that you're seeing the provost heading to his ride. So I'm happy to introduce Provost Andrew Rogerson. Thanks.
Wow, that's quite a build-up. I'm a bit scared to be up here now. Well, I know this has been a whirlwind of a course. Now we're on to a completely different subject for you, but there is a constant theme I hope you can see, which of course is change. In my case, I'm very aware of the limitations of trying to lecture in this space, so I will be using PowerPoint. The lettering will be larger than you see at the moment, so don't panic. I also try to put as little as possible on the PowerPoint so that taking notes is really a question of key phrases or even just some single words. The view to this is not to try and make you scientists in 5 hours, because we're going to be talking quite a bit of microbiology. And it's not to make you a microbiologist, because some of you actually have probably got quite a good background in micro, and some of you are probably in the humanities. So everyone will be at the same advantage, so to speak, because you're not being made into microbiologists. I'm just hoping to open your eyes to just how powerful science has become in recent years. And everyone here has to really pay attention to that fact, because you'll all be called upon to make decisions as you go through your life, decisions about what scientists are now capable of doing. And so that gets you right into the face of ethical conflicts. So that's what the course is really about. It's about making you aware of the power of science, the changes that have happened very recently and very fast, and how that gets you into an arena where you have to make some decisions. So you have to be informed as you go forward in your lives. What you're looking at there on the screen is, it's the sort of part that you're probably most wanting to know. How do I pass this piece of the course? I'll be giving a multiple choice exam. I think it's on the 29th of April. It will be short, maybe 20, 25 questions. The content will be based on everything that we talk about in the next 5 hours in this room, and it will also cover these readings, which you can find. They're all from the UNESCO Courier, which is the United Nations Education and Culture Organization. You find them by clicking on that website, and then you look at the various, let's see if I can get this right. There are several readings there. You find them by simply keying in the name of the topic, the name of the article, and then it will take you right to it. Don't be horrified. These are very short articles. In total, this is 31 pages of reading, so it's very light, it's very easy, and it will supplement what we talk about in class. So everything we talk about is examinable. Everything in those 31 pages is examinable, but it's all based on trying to reinforce and get across the meaning I hope to convey to you about what has ethics got to do with microbiology. Is that clear, everybody? We're good? I also hope, and this bit is just, would be my dream, that as we talk about some key phrases as we go along, or you come upon a word that you're not familiar with, or you hear about Escherichia coli, which is a bacterium, and it excites you, you go and you look for it and get some additional reading on it just off the web. Everything we talk about here is incredibly easy to get extra information to reinforce, so there's no textbook or anything else recommended. It's going to be very easy for you to get additional information if you get charged up about what you hear. The first question then is really, what is ethics and microbiology? Why are they linked together? Why should microbiologists even worry about ethics? And of course, the problem is that over the last 25 years, we've come to understand so much more about microbiology, and it's given us some rather strange abilities. It's given us the tools to be able to start manipulating organisms. And one thing I want to get across to the group is that the reason that we can do these manipulations now, in other words, we can change how organisms work, is because of microbiology. All the tools that you get to be able to do this come from microbiology. So that's why we're going to be talking quite a bit about microbiology, so as that makes sense to you. Of course, the concern is that now we've got the ability to do some fairly dangerous things. I mean, people out there are saying that microbiologists now, rank in the realms of a nuclear engineer in terms of the capability to do harm. It's true, we could make a superbug that could kill half the population of the world. But on the other hand, if we don't worry about doomsday bugs, which we hopefully will never manufacture, we're also able to do incredible good with this kind of manipulation. The viral disease polio, which was once a scourge of the planet, has all but been eradicated. And that's because of vaccinations, and that's because that's a tool that we've managed to get from microbiology. This was a disease that you used to get from sewage-contaminated water. And as I say, it's all but eradicated from the planet. Others out there, when they see what microbiologists can do nowadays, complain that we've got too much power and we've been accused of, say, playing God. And that concerns a lot of people out there. What we're really talking about here is genetic engineering, and we'll clarify what that means in a moment. And the other concern from people is that genetic engineering is being used across the planet to make incredible sums of money for big business, because many of these innovations are made by the big companies, and when money drives the issue, then sometimes money gets in the way of making the right decision. And again, that's something we're going to be touching on. But let's be clear, because I'm a microbiologist, I have to stand up for microbiology. Microbiology has allowed us to do many good things. It's allowed us to make vaccines of the kind I just talked about to eradicate polio. It's allowed us to use what are called viral vectors to move genes around. So we'll clarify what number 2 is all about. But vectors is a word you're going to hear several times in the next 5 hours. It, of course, allows us to make food and beverages. Anyone here that enjoys a glass of beer or wine, or are going to enjoy a glass of beer or wine when you're old enough, should at least celebrate microbes, because the reason we can ferment is because of microbiology. We're also able to make new organisms nowadays to make useful products. And that, of course, is what genetic engineering is all about. And we'll be talking about that to clarify it. And we can also make organisms now that do what we call remediation. That's the ability to take an organism, to take contaminated ground, and then clean it up. And that can be done by making an organism that can live in a very contaminated environment and use that contaminant, and in using it, it breaks it down and cleans up the area. That's called bioremediation. Remediation is a big topic these days. And if you get nothing else out of this course, I hope you're going to accept this statement that's made by one of my favorite educators on the planet, and that's Eleanor Siebert. And she says that the greatest reward for all of us in science education is facilitating the development of able and ethical scientists, as well as a science-literate public who can support and judge their work. So she's saying that we want more and more people to go into science and become those scientific leaders that we urgently need on this planet. But she's also saying that everyone should become familiar with science so that you can be the controls, you can judge what's right and what's wrong, what is ethical. So let's start then by turning you into a microbiologist. In a very simple sense, let's be very clear about it. Microbes are defined as any single cell that can do its entire growth as a single cell. It sometimes can form clusters like these, but they're still individual cells, and by definition they are called microbes or microorganisms. Makes them very distinct from everything else on the planet, like plants and animals and people, which are all multicellular. They're also incredibly small. We are familiar with what a millimeter looks like on a ruler. A microbe is usually well less than a millimeter in size. Most of the microbes are about 1/1000th of a millimeter, which takes it to a unit that we refer to as a micron. So a micron is 1/1000th of a millimeter, and because microbes are so small, we tend to talk about microbes in terms of microns. Of course, because you're down at that size, almost every microbe on the planet is less than 0.5 millimeter or less than 500 microns, then they're all invisible. So very few of them can you actually see with the naked eye. They come in many shapes and sizes, and I'll just give you some names that you're familiar with and give you a feel for what they look like. The absolute smallest, of course, are the viruses. These are well short of even being 1 micron. The largest virus is about 1/10 of a micron. And these are truly invisible. It's also sort of incorrect to call them microbes because a virus cannot reproduce by itself. So it doesn't really form the definition of a single cell. So it's more correct to actually refer to viruses as viral particles rather than viral microbes. But just be aware of that. We'll talk more about viruses later. Another microbe that everyone's heard of course, are the bacteria. Bacteria are about 1 micron in size, very small, but that's about 1/10,000th of a 1/1000th rather of a millimeter. They're very unspectacular to look at. This is them stained under a microscope. They just look like little dots or little rods, or sometimes you get excited and you see a bent one. And that's about as exciting as bacteria get to look at. But there are thousands and thousands and thousands of different kinds of bacteria, and we'll touch on that later. Another group of microbes that are single cells are the algae. These are ones which are green. They can photosynthesize. They can turn the water green. You can look at the pond on the campus, and sometimes it's a nice green color, thanks to all the algae growing in it. But these, because they are single cells, are microbes. And then you come to a whole group of microbes that eat things, and they all come under the heading of protozoa. So protozoa are the consumers of the microbial world, and they are themselves, of course, microbes. These guys, you can hardly see them, they're called little flagellates. Flagellate written there, because they've got a little whipping flagella, which is how they swim around. These are really interesting, and we could go off on a million digressions here, but up until about 10 years ago, we had absolutely no idea that these were very common. But it turns out that in every drop of water, take a teaspoon, there are 1,000 of these swimming around, and yet very difficult to see. And there's 1,000 of them because they're all busy eating all the bacteria that are in that drop of water. And it's because of these little grazers that the world isn't overrun with bacteria. Bacteria divide very fast, and if nothing was eating them, we'd soon be walking across a soup of bacteria. But these are one of the major consumers that keep the bacteria out in the natural world in check. These are also consumers. They're called ciliates because they've got little hair-like cilia, and they also eat other microbes. And then have to include the naked amoeba here that John talked about. These are the kind of amoeboid organisms that flow around and engulf prey. Don't worry about any of this as detail. Just be aware that there's a lot of diversity from viruses, bacteria, algae, protozoa. They're all microbes. So why would we bother studying microbiology? If I had an interactive class, which I know you can't be in this hall, I would ask, why do you think we want to study microbes? And I imagine most of you would turn around and say, because they make you sick. And you would be correct. There are some microbes that can make you quite ill. And so that's a good reason to study them. For instance, you've all heard of Ebola. That's a very significant disease that is caused by a virus. Ebola can kill you within 24 hours with hemorrhagic fever. You literally dissolve basically from the inside. Your whole body starts to bleed internally and you're dead. And Ebola, thankfully, can be detected very fast because when someone gets sick with Ebola, you know they are sick because they're basically dying. And that is a salvation for all of us, because imagine the situation if we had some disease like Ebola that didn't show the symptoms until, say, 2 weeks later. That person would have infected many, many people. And of course, this is the scare of these emerging diseases, that we get a disease that comes along that is lethal as Ebola, but doesn't kill you very fast. Because it kills fast, it can be contained. Ebola, by the way, was named after the place in Africa where it was discovered, the Ebola River. And then we've got bacteria making you sick. I think everyone here has heard of Escherichia coli, which is a bacterium that every one of you has in your gut right now. And should that bacterium get on someone's food, it can give you a pretty unpleasant bout of food poisoning. It's a good reason why you want to cook those hamburgers fairly well, because if meat gets contaminated with some E. coli on the outside, it's not a problem if it's a steak, because you just fry it up and anything on the outside is killed. But think about hamburger. It's all ground up so you can move that bacterium into the middle, and then you eat that nice pink fleshy meat in the middle and you get sick as a dog. So be careful with E. coli in hamburgers. They can make you ill if you don't cook meat well enough. And then another one you might have heard of is Campylobacter. You don't need to remember these names. But that's the commonest cause of food poisoning if you eat chicken and other poultry. So we could study microbes because of disease. We could also worry about the other kinds of microbes. I don't want everyone to think you only get sick from viruses and bacteria. Protozoa can make you sick. If you ever go and drink water, particularly in Mexico, it's a problem down there with the contamination. You can get very sick with entamoeba. That's quite common, drinking polluted water. And then algae as well. I don't know if everyone here has heard of red tides, but we'll maybe come back to that topic later on in our 5 hours. A red tide is an explosion in the water of this type of alga that becomes so abundant it forms one of those blooms, but instead of turning the water green, it turns it a reddish color because this particular dinoflagellate has a red pigment in it. And the danger there is if you eat fish or shellfish that's been contaminated with this particular alga, you can get very sick. In fact, you can die. You can die of something called paralytic shellfish poisoning. Some of you might have heard of that. If you're ever eating shellfish and you find a tingling on your tongue, don't eat any more. It's probably got that toxin in it, which could be very dangerous. It's called saxitoxin. It's not something you should be that concerned about. It only kills about 5 people a year, but you don't want to be one of those 5. Tingling from eating seafood, don't eat any more of it. So, we could study them because they make you sick, but that's really a bad reason, because way, way less than 1% of microbes causes any concern in terms of health. Tiny number cause you any concern. If I had to choose something, I would say we study them because they're so incredibly abundant. Let's think about it. It's the age of bacteria. Don't be disappointed. I know you all think it's the age of man or woman. Not true. Bacteria were on this planet first. Bacteria dominate the planet now. And when we're all extinct and we've destroyed the planet with whatever we're going to do to it, sorry to be so cheery, bacteria will still be there. So it is the age of bacteria. And look at the incredible numbers of them. Every one of you sitting there has 4 pounds of bacteria in and on your body. Yep, it's actually 4.2 pounds. Your gut is full of them, your skin is covered in them. And if you were to remove them all, you would lose 4 pounds of weight. You'd also get quite sick because you're in balance with them. They do you some good as well as trying to do you some harm. Your skin, as you see, has 100,000 of them in a square centimeter. A teaspoonful of soil from just outside, 10 trillion, spelt wrong, bacteria. And if you wanted to know how many bacteria there are on the planet, it's been estimated as 5 million trillion. And because that means absolutely nothing to you, look at it with all those zeros. That's a 5 with 30 zeros after it. That's enough bacteria. If you took them all, it would cover France to the depth of 3 feet. Think about it. That's using microbes that are so small they're invisible to the naked eye. Another reason we might want to study them is because of diversity. They're everywhere, like we've just said, in terms of numbers, but they're everywhere wherever there is water found. They're also incredibly important because although they may all look the same, I just told you bacteria basically look like little dots or little rods, but their diversity, their ability to do things on the planet is enormous. And so they do important functions like recycle nutrients. Nitrogen, for instance, in case you hadn't ever thought about it. If we didn't have microbes, there would be no nitrogen going into the soil, there'd be no plants, there'd be no life. It's bacteria that recycle materials, break things down, and bring them back so that other organisms can use them. And of course, this amazing diversity of different ways of eking out a life on the planet gives us the opportunity to use them in very unusual ways for our benefit. And that gets you into this term biotechnology. It's because they are so amazing in their array of different ways of performing their function on the planet that allows us to say, aha, that's a useful product. That's a useful way of living. I'm going to use that microbe in a biotechnological way. The definition of biotechnology, seeing we're talking about the subject, is the use of microorganisms or biological substances, such as an enzyme, which would come from a microorganism, to perform industrial or manufacturing processes. I know it's a mouthful, it's get your head around it, but all we're saying is it's use a microbe in any application you can think of, and if it does something useful, that is biotechnology. And so the reason we have these wondrous things like biological washing powders is because of microbes. Cold water washing powders. Think about it. That's because they have taken an enzyme which can break down grease and dirt, and they've taken it from an organism that lives in cold climates or cold water. So when they say it's a cold water biological washing powder, they mean it. Don't use hot water, you'll kill it. The enzyme will break down the dirt on your clothes if you use it in cold water. It came from an organism that lives in cold places. We've talked about fermentation products already. Oops. The fact you can make beer and wine. It's because of microbes. We've talked about genetic engineering, or we've alluded to it, and we'll talk more in detail. Genetic engineering is, of course, the ability to move the genes around from one organism to another organism, to engineer the genetic material, genetic engineering. And of course, we also mentioned bioremediation. This is the idea that we can manufacture an organism that can then be used to clean up the environmental damage that we've been so good at doing on this planet. We have sites where, for instance, the soil creosote, which used to be used to preserve wood. There's so much creosote has got into that soil. And we put a fence around it, and we just have to look at it. And the great promise is we could make microbes that could be thrown in there, and they could do their magic and eat up the creosote. What's the danger with doing that? Why haven't we done it yet? Well, we've got the capability, but we're a little scared about what happens if you release all those microbes out into the natural world. Where are they going to go? How are we going to contain them? How are we going to prevent them changing into something else? So there is a lot of concern about releasing genetically engineered organisms. We'll come back to that. That's a very ethical issue. But be very clear, there's a lot of pressure coming from big business to start using some of these microbes we know could clean up oil spills or could clean up creosote soil. These sites, just so you are all aware of it, with a fence around them and contaminated soil that we can't go near, are called Superfund sites. They're called EPA Superfund sites, Environmental Protection Agency Superfund site. If you've never heard of Superfund sites, I encourage you to go online and type in EPA Superfund, and it will tell you how many Superfund sites are in your neighborhood. You might be surprised. Anyone that's thinking of buying a house, I strongly suggest you go on and check for Superfund sites. You don't really want to be living next door to one. I checked yesterday before I came to this lecture and put in Sonoma County. Sonoma County is a pristine environment. You wouldn't expect to have any Superfund sites. We have 63 in Sonoma County. So check it for your home area. You might be surprised. Of course, some Superfund sites are small. Gas station that's leaked gasoline into the soil could be a Superfund site because we cannot clean up that soil. It doesn't always have to be hundreds of acres. But we have 63 of them in Sonoma County. Anyway, back to tools from microbes, because the whole point of this microbiology lecture, because it's turning into a microbiology lecture, is to try and open your eyes to what we're getting from microbes that has allowed us to play God, if you want to go with that phrase. One of the most famous tools came from a bacterium that you can find in a hot spring. The bacterium is called Thermus aquaticus. Thermus means warm. Aquaticus means it came from warm water. This one was found back in the 60s. Actually, sorry, back in probably later than that, in the 70s. And was really what started to make biotechnology possible. This was a huge find. The product from it that made the finder of this so much money is because he isolated an enzyme from it. And that enzyme is called Taq polymerase. Any enzyme always ends in ASE at the end. So if you're a protease, you're an enzyme that breaks down protein. If you're a polymerase, you're an enzyme that, in this case, helps you polymerize something, which is join it together. But we're getting into too much detail. This is a very important enzyme, and we'll talk about it later on. As you see there, the definition of it is it's an enzyme that makes DNA join together or replicate. And of course, it's the basis of a multi-million-dollar industry which uses something called PCR, or polymerase chain reaction technology. Throwing some titles at you or topics at you which some of you will be confused about, some of you will have heard of PCR, but we'll clarify all later. Just put it at the back of your minds. Taq polymerase is a very important enzyme coming from microbes that give us the ability to do something called PCR, polymerase chain reaction. Big bucks. It's why if you go to Yellowstone today and you try scooping up some of the water from the hot springs into a bottle, you will be in serious trouble. They don't let you collect water from hot springs anymore because they don't want you finding valuable new microbes that could end up doing something like Taq polymerase. It's illegal to collect water in federal parks now because of Taq polymerase. The park would rather make the money than let you run off with your new microbe in the bottle that you're going to look for. It's a different world now because of microbiology and biotechnology. Because we're talking about change, I think it's relevant that we educate you a little bit about some of the pioneers in microbiology, because it's all good fun to see how microbiology came about. And they're not in any numerical order or even by chronicle order, by date, but one of the first important microbiologist, if you like, as far as I'm concerned, the most important, was Anton van Leeuwenhoek. In 1676, he was the first person to describe and see a microbe. Remember, they're invisible, so you need a microscope. And Anton van Leeuwenhoek was a Dutch merchant who had a hobby, which was making microscopes. And in his spare time, he'd look at rainwater and pond water, and when he did that, using his very crude microscope, he would see microbes. And he published it. Way back then, in the Royal Proceedings of London, he published pictures of microbes. He was the first person to see protozoa. He was the first person to see bacteria. So Anton van Leeuwenhoek was the man who saw microbes. He wasn't the man to invent the microscope. We actually had a microscope before then, which was invented, but that person who invented that microscope didn't actually look at microbes. They looked at cells. So that's a little piece of trivia. Do you know why, when he looked at, it was actually some cork tissue, he could see cells and named them cells? I love this interactive class because I can ask questions and I don't really expect anyone to answer. But I'll tell you, he named them cells because it reminded him of the cells in a monastery. All these little regular rooms that you have the monks in a monastery are known as cells. And when he looked at tissue under the microscope, it resembled cells to him. So that's how we get the name cell. But he wasn't the father of microbiology, because Anton van Leeuwenhoek looked at individual cells, single cells, microbes. We also have to put up Hippocrates in this list because way back in 400 BC, think about it, that's 400 years before the birth of Christ, Hippocrates was able to anticipate microbiology is probably the best way to look at it. In other words, he set the standards for medicine that we still use today. If anyone's going into the medical profession, you will use some of the practices that Hippocrates set out. Things like, if you are sick, then isolate the sick person from people who are not sick. That may seem awfully obvious to everyone in this room, but in 400 BC it was brilliant. And of course, by moving people away who were sick and isolating them, you are then preventing the transmission of disease. And in that way, he anticipated there were microbes out there, although of course he had no idea that there were such a thing as a microbe. But he knew he had to get those people away from healthy people. Another one that we should pay attention to is Fracastoro. In 1546, this Italian physician actually suggested that there were invisible microbes causing disease. Can you imagine in 1546 standing up and telling people you're getting sick because of something we can't see that's a little infective agent? Brave man. Of course, no one believed him because no one could see these particular organisms. Another one is Edward Jenner, who's in 1798, and he was the first scientist to be able to come up with a vaccination that worked. And Edward Jenner is an interesting man because he got this idea by following his father around. His father was a vet and used to go out to a lot of farms. And at the time, in the 1700s in England, smallpox was a very, very serious disease that was killing thousands and thousands of people. And Edward Jenner noticed that the milkmaids who would take the milk from the cows always had fair skin and never ever came down with smallpox. Smallpox, of course, is a very disfiguring disease, so you can immediately tell when someone has got it, or if they were lucky enough to survive, someone who had it because their face is covered in pox marks. Milkmaids never ever had smallpox, and this fascinated Edward Jenner. And he made the connection that cowpox, which was a disease that cows had, is a very similar disease to smallpox, except it doesn't disfigure and it doesn't kill. So it's a much sort of less virulent, is the correct way to put it, form of the disease, smallpox and cowpox. And so he managed to make a vaccine based on cowpox, which was not damaging, but was enough to act as a preventative vaccine against smallpox. He injected 25 people with it, and it gave them, all 25 of them, protection to the much more virulent disease, smallpox. And so that was the first vaccination. And again, I'd love to interact with this class, and I'll just ask a question. You can all answer it inwardly. Why is a vaccination? Why did it come up with the name? What is the Latin for cow? And you're all saying to yourself, vacca, and you're correct. And of course, vacca is the Latin for cow and is the derivation of the name vaccination. Bit of trivia for you. We also have to include Louis Pasteur in this. He was a chemist, a French chemist in 1861, and he's done many things, of course, one thing being pasteurization. You can thank Louis Pasteur for the fact we can now pasteurize milk and other dairy products. Pasteurization just means you're removing most of the organisms that could make you sick. We don't need to get into the details. But he did a lot more than invent pasteurization. His biggest claim to fame, I believe, is he ended a belief in spontaneous generation, because in the 1800s, it was the view that if you left out a piece of meat on the side of the kitchen table for a few days, you'd come back and it would, of course, go rancid. And it would get very smelly. It'd get contaminated, of course, with bacteria that we know about, but they didn't know about that at the time. And eventually, if you leave it long enough, you'll see maggots crawling out of it. And the belief was that those maggots came from spontaneous generation, because no one else could really understand how the maggots got there. So they came out of what was called the vital force of the meat, enabled these things to generate spontaneously. I know it sounds ridiculous, but that's what people believed in the 1800s. And of course, Pasteur, with a series of elegant experiments, showed that that was absolute rubbish, and that is, I believe, his claim to fame. Joseph Lister, I have to add in there because you've all heard of the product that bears his name. He was the father of antiseptic medicine. I know it seems crazy that doctors used to go in and do an operation, saw off someone's leg in the 1800s, and immediately go and then help deliver a baby without even washing their hands. It took Joseph Lister his entire life to be able to convince hospitals that they have to start practicing antiseptic medicine. Washing your hands in carbolic soap is all it takes, and the death rate in hospitals went down. It took him his entire lifespan to get that message across. His claim to fame, of course, is Listerine that you can use as a mouthwash, which of course had nothing to do with Joseph Lister, but it is an antiseptic you can swirl around your gums. And of course, I have to add Alexander Fleming to the end, who is the discoverer of penicillin. I like to put him in because he's Scottish, and in case you hadn't caught on, I'm Scottish as well, once upon a time. But Alexander Fleming, of course, discovered penicillin when he was looking at a petri dish and saw there was a fungus growing in it called penicillin. And of course, around that fungus, there was no bacterial growth. The fungus was giving out a product that was inhibiting the growth of bacteria, and that product, that antimicrobial product, became the first drug, penicillin. And of course, it was then manufactured in the war in the 40s and saved countless lives, and has continued to save countless lives. And of course, now we've added many more antibiotics to the list of penicillin. So that's just a kind of flavor of some of the landmarks. And we'll just continue by talking a little bit more about some names in microbiology you need to at least be aware of. In this case, structures, not so much names, so that we can feel more comfortable as we continue the discussion about what did those microbes give us that allows us to do all this wondrous biotechnology. Again, do not go home and memorize any of this. I'm not trying to make you scientists. The only name you have to take away from this is this piece here, eukaryotic cell. So some cells are very complicated, and these are all the cells of multicellular organisms, like yourselves. If you were to take one of your cells and chop it open and look at it with a microscope, you would see it's got lots of structure inside, and you don't need to know about any of those structures, except they're all very organized, they're all in membrane-bound structures, and the DNA, for instance, is in a round structure called the nucleus. So this is a very complex type of arrangement, but this is a eukaryotic cell. The difference is with the microbes, or at least the microbes in the bacteria class, which is this huge, huge number of diverse, abundant microbes, they're very much simpler in terms of their structure. And the only word you have to take away here is prokaryotic cell. So bacteria have got a prokaryotic structure. Everything else in the planet has got a eukaryotic structure. That is the main difference between bacteria and everything else. The lack of structure in a prokaryotic cell means that the DNA is not organized in a nucleus, it's just scattered around the cell. So that green smudge, which you could probably barely see at the back of the room, apologies as always, that green smudge is just a big circular link of DNA. The relevance of this is if you want to go in and make changes, it's so much easier to do it using bacteria. Think about it, to go in and play around with the DNA when it's just all out there and naked is a lot easier than going in and playing around with the DNA in a very organized cell. The other reason for telling you about prokaryotes is many of them, and apologies, I just grabbed these and I don't know how to do PowerPoint anymore. It's been so long since I had to lecture, and I didn't know how to get the whole picture on the slide. But that word there that's cut off is plasmid. And you do need to know what a plasmid is. P-L-A-S-M-I-D, plasmid. Now, this is another crude diagram of a prokaryotic bacterium. And you can see the DNA that I talked about, the large circular chromosome. But a lot of bacteria also have these circular pieces of DNA called plasmids. Okay? So what you've had to take away from this is we have eukaryotic cells, which are very complicated and very organized. You have bacteria, which are all prokaryotic cells. And some of those bacteria have plasmids. Plasmids are really interesting because they give an organism some unusual characteristics. The reason that bacteria are antibiotic resistant, in other words, as we all know, because we're continually challenging our microbes that are trying to make us sick with antibiotics like penicillin, these bacteria can cotton on fast and change. And they can change and become what's known as antibiotic-resistant bacteria. And these are very worrying. And usually that means that they have a gene, and it's usually on the plasmid that allows them to be resistant. Okay, let's just be clear about this. So a gene is a segment of DNA. And it's made up of all these different bases that we hear about. Bases making up a gene in amongst the whole strand of DNA. And that area of gene makes a protein. Okay? We have thousands and thousands of genes in our body, all making different proteins. So genes make proteins. And proteins are important because they are the messengers of your body, they are the structural components of your body, they are the components that make your body do things. Hormones are proteins. Enzymes that break down food are proteins. And other agents in your body like muscle tissue is a protein that gives you structure. So it makes sense then if you're trying to control what an organism does, you do it by the different kinds of proteins that organism makes. And if you want to destroy an antibiotic that's flooding into your single cell, the way to do it is to make a protein that breaks down penicillin. And guess what that would be called? I told you earlier, all enzymes end in ASE. So that bacterium has a gene that makes penicillinase. And that gives it resistance to penicillin. So that when you get sick with that organism and the doctor gives you penicillin, it doesn't work. Because as soon as that bacterium sees penicillin, it breaks it down with its enzyme, and suddenly it divides and divides and divides, and you get very sick. So that's the big scare, that the more we challenge microbes, the more they rise to the occasion and beat us. And we are now seeing endless amounts of examples of antibiotic-resistant bacteria. There are some strains of gonorrhea you can catch now that you cannot get rid of because we don't have any drugs that can treat it. Don't mean to be overly dramatic. I could have chosen any disease, but that's just out there. And usually then, the relevant part is those important genes that confer important conditions, in this case antibiotic resistance, are usually found on plasmids. And as we're going to hear, the thing about plasmids is they can move around. You can go and a bacterium can, this plasmid can come out of a bacterium and go into another bacterium, completely different bacterium, completely different species of bacterium, and lo and behold, suddenly that other one has got antibiotic resistance. So plasmids move around a lot in nature. Plasmids can be easily moved around by microbiologists. And so plasmids are important because they are vectors in genetic engineering. Okay? That's the one word you have to take away from today's lecture. Vector. Vector is the means, the tool for moving genetic material from one organism to another. We can use plasmids as that tool. How are we doing? Information overload, or are we still hanging in there? Information overload, I like it. I have been told I got to let you away about 22, so you will get out on time. We're going to have another 5-10 minutes at the most. Give yourselves a shake. You're doing good. Just remember as well, microbes divide pretty fast. Another reason you can get sick pretty easily with them if you take your hamburger that you haven't cooked very well and it's been contaminated with Escherichia coli, and that is sitting out in a nice warm grill for the afternoon before you get round to barbecuing it. E. coli can divide every 15 minutes. So that means that one single invisible E. coli sitting in the middle of your hamburger, in 12 hours, it makes over 6 billion E. coli. That's almost more than the population of humans on the planet. In 12 hours, one E. coli can become 12 billion. That's also important in terms of that ability to survive antibiotics. It doesn't take very long for that one organism with bacteria, with antimicrobial resistance, to suddenly dominate the entire population that's invaded your body, because they can reproduce so fast. Okay, one other vector we're going to talk about. So plasmids, one important vector from microbiology. Let's just make sure you all know what a virus is, because viruses are also important vectors. Remember, we're talking about the ability to move a piece of DNA from one organism to another would be called a vector. It happens in nature by itself, but it happens in the laboratory by genetic engineers. Peter Medawar was a Nobel laureate way back in the 1960s. He's an immunologist and he got the Nobel Prize for it. And I just love the way he describes a virus. It's a bad piece of news wrapped up in a protein. That's all a virus is. So a virus is not a single cell. Remember, we said that they're not really microbes. We tend to call them viral particles if you want to be really correct. And they are just simply, as Peter Medawar said, a piece of DNA, and on the outside it's encased in some kind of protein. And we fear them because viruses can make us really sick, and whether you realize it or not, we're actually pretty bad at curing any viral diseases. We haven't even been able to cure the human cold. We can't cure cold sores on your lip from herpes virus, no cure whatsoever. We can't cure viral diseases such as AIDS, and we certainly can't cure viral diseases such as Ebola. So think about it. Although we've made great advances in medicine, we're only really good at treating bacterial diseases. Viruses are really difficult. They're a bad piece of news. It's also the reason that the name virus was chosen because, again, anyone here that knows Latin, which is probably none of you nowadays because you're so lucky you didn't have to do it like I had to do at school, but a virus actually comes from the Latin meaning poison. So, yes, we fear viruses because they are poisonous to us and very often kill us. As I said, they are very small and they cannot replicate on their own, so we call them particles. So they are neither prokaryotic or eukaryotic. They are not cells, so we can't call them either of those two. They are intracellular parasites. And they're very, very small. The largest of them is about 0.1 of a micron, which we said earlier on. So here we have something that's the ultimate parasite. It comes along, it lands on one of your cells, it has this wonderful way of then injecting itself or its DNA into your cell. It then takes over your cell's genetic material and it tells your cell to make new viruses. Don't make a new skin cell. The viral DNA goes in, takes over your machinery, and says, this skin cell will now make new viruses. It's why most viral diseases are manifested by unattractive lesions. Because once you've been infected and your cells have replicated new viruses, they have to get out. And so they burst open the cell, and out come all these viruses. A cold sore on your lip is just the result of all your lip cells bursting open to release all those viruses. Should you go and kiss someone who's got a cold sore, those viruses will then attack your lips. And of course, that's how they get transmitted. They're a nasty piece of work. And that's a good place to leave this lecture. See you Monday.